Hello and welcome back, everyone. So this is the week 4 lecture, and we will be going over muscle mechanics this week. When we look at this slide over here, we see that at all the way on top we have the level of the entire muscle. This is the muscle level. As we come down, we see that muscles are made up of fascicles. Well, what are fascicles made up of? They're actually made up of muscle fibers, or the muscle cells. Well, what are these muscle cells made up of? They're made up of a sarcolemma, T-tubules, sarcoplasm, and they have a lot of nuclei. And there's other stuff also, but let's just stick with what's written over here. So again, they're saying that the transverse tubule and the sarcolemma, they're together. They're actually one structure. They end up turning into a single structure, right? The transverse tubules, they're actually invaginations of the sarcolemma. So now when we come over here, so what about the sarcoplasm? What do we get from the sarcoplasm? Well, think of the sarcoplasm as the fluid, the area that's inside, that's surrounded, that's enclosed within the sarcolemma. And what we find is that it's made up of a sarcoplasmic reticulum. Okay, and again, you will see that these transverse tubules are also connected to the sarcoplasmic reticulum. They function together. They're functionally linked. There's also mitochondria that we find there. And also we find glycogen granules, which is an energy source. So what about the myofibrils themselves? What are their structural building blocks? And we see that they're made up of the protein troponin, actin, tropomyosin, myosin, titin, nebulin. Well, what about myosin? Myosin ends up making thick filament, and actin ends up making thin filament. So when you look at the organization of these thick and thin filaments, they get organized into sections that we call sarcomeres. So looking over here, we see a sarcomere. Again, a sarcomere goes from a Z disc to a Z disc. Okay, this is a continuation from the previous lecture that we had last week. And then over here you can see that thin filaments and thick filaments that we have over here. Okay, and then you can see the structural proteins. This would be titin, for example. This is the Z disc over here. And so again, your thick filament and thin filament. And we said in the previous lecture, thin filaments, the building blocks are G-actin molecules, which assemble to form the F-actin strands. Okay, myosin is forming. It's what's forming this M line over here. And then we have the titin proteins over here. Okay. So what is a cross bridge? Well, we said that the cross bridge was when the myosin heads would attach to the actin filaments. This is the simplest way that we can describe what the cross bridge is. And remember, these are microscopic structures. You can't see it with your naked eye. If you were to look at a muscle, you wouldn't be able to see it unless you look at it under a microscope. Some questions to explore. The first one, how does an action potential trigger the contractile process? Next one, what's the source of the calcium? And what's the function of the calcium? Again, go back to the previous lecture and you should be able to answer these. And this slide is also a review of the previous week's material. So again, what they're saying is that before the contraction begins, we need to have energy. So ATP molecule needs to bind to the myosin heads. So once ATP binds to the myosin heads, on the myosin heads, remember, there's the ATP binding site, and within there, it has the capacity to hydrolyze the ATP, to break down the ATP. So again, it has ATPase activity. So that ATP ends up becoming ADP, adenosine diphosphate, plus phosphate, and it stays connected to the myosin head. So when the troponin binds with the calcium, then the tropomyosin will be moved off of the the myosin binding sites on the actin, on the G-actin filament. At that point, now the myosin head is able to bind to that G-actin molecule. So after we have that cross-bridge formation, the head of the myosin is going to end up changing shape. It's going to cock inwards. And when it cocks inwards, what's going to end up happening is we end up having the thin filament that gets pulled in. So this is what we call the power stroke. Okay. So once that head tilts, now the ADP and phosphate ions, they're able to separate. It will be released. Okay. Once it's released, that myosin head, it stays binded. It doesn't let go. It continues to be binded to the actin molecule. Okay, it's waiting for another ATP to come in, and once another ATP comes in, a new ATP comes in and attaches to that the ATP binding site on the myosin head, now it will detach from the G-actin molecule, and then this thing is going to end up happening over and over again. Okay, the cycle will repeat. So the last thing that it says that the cycle of cross bridges, binding, bending, and attachment complete the shortening and contraction of the muscle. So again, this just keeps happening over and over again. Once again, going over the sliding filament theory of muscle contraction. So in the sarcomere, the basic unit of muscle contraction are the thick filaments and the thin filaments. Okay, so the thick filaments are made up of myosin and the thin filaments are made up of actin. Okay, we also find trypomyosin and troponin, right? All these things are arranged in a specific pattern. So during muscle contraction, the myosin heads on the thick filaments, they attach to the actin on the thin filaments, as we have discussed in the previous slide. And then the thin filaments are pulled, okay? And they get pulled towards the center of the sarcomere. This is what we call this power stroke. All right, now, keep in mind that the thick and the thin filaments themselves, they don't actually shorten during muscle contraction. Rather, it's the length of the sarcomere as a whole that shortens due to the sliding of the thin filaments past one another. So a single muscle twitch refers to a brief, sudden contraction of a muscle that occurs in response to a single, short-lived electrical stimulus. Now, this stimulus can be delivered to the muscle directly, or it can come from a nerve that innervates that muscle. So during a muscle twitch, there's a sudden increase in the electrical activity within the muscle fibers, which leads to the release of calcium ions from the sarcoplasmic reticulum. These calcium ions, then they bind to troponin, and remember, troponin is a protein that we find on the thin filaments of the sarcomere. This triggers the sliding of the thin and the thick filaments past each other and the subsequent contractions of the muscle. Now, the duration of a single muscle twitch is very brief, lasting only a fraction of a second. However, the strength of the contraction can vary depending on the size of the stimulus and the number of muscle fibers that are activated. If a larger stimulus is delivered to the muscle, or if more muscle fibers are activated, a more forceful contraction can occur. All right, so muscle contraction can be classified into two main types, isometric and isotonic. So isometric contraction occurs when the muscle contracts, but it does not change in length. During this type of contraction, the tension within the muscle increases, but the muscle doesn't produce any movement. So a good example of this would be when you try to lift a heavy object that's too heavy for you to actually lift. And what's happening here is that your muscles will contract isometrically to try to lift the object, but since you're unable to actually move that object, your muscles don't change in length. Now, when we contrast this to isotonic contraction occurs when the muscle changes in length while maintaining a constant tension. And a good example of isotonic contraction is when you lift a weight during weightlifting. So as you lift the weight, your muscles will contract concentrically, shortening and producing tension to lift that weight. As you lower the weight back down, your muscles will contract eccentrically, lengthening while still producing tension to control the movement. Alright, so once again, isometric contraction can occur when the muscle contracts, but it does not change in length. Now, isotonic contraction occurs when the muscle changes in length while maintaining a constant tension. And this type of contraction can be divided into two subcategories, eccentric and concentric. So, concentric contraction occurs when the muscle shortens while producing tension, while eccentric contraction occurs when the muscle lengthens while producing tension. Okay, so concentric, muscle shortens; eccentric, muscle lengthens. So if you want to look at, think about other examples, okay, of isometric and eccentric and concentric contractions, right? And you should write them down, list down examples of a few, aside from the ones that I've given. What else are examples of concentric contraction? What else are examples of eccentric contraction? How about isometric contraction? Okay, can you give some examples? All right, so let's talk about the characteristics of whole muscle contraction. So the characteristics of isometric twitches that are recorded from different muscles can vary due to the differences in size, diameter, and the energetics of muscle contraction amongst the different skeletal muscles in the human body. Now, isometric twitches are brief. They're sudden contractions of a muscle that don't result in any movement of the muscle. They're typically recorded by measuring the force that's generated by the muscle during the twitch. The mechanical characteristics of muscle contraction can also differ among muscles due to their different size and diameter. Okay? So, for example, larger muscles may have more muscle fibers, and thus they're capable of generating greater force than smaller muscles are. Okay, each motor neuron that leaves a spinal cord in the human body is responsible for innervating multiple muscle fibers, with the number of muscle fibers innervated depending on the type of the muscle. So the group of muscle fibers innervated by a single motor neuron is called a motor unit. Okay, so we have a motor unit here, another motor unit here, another motor unit here, and another motor unit here. Okay, because why? Because we said that the group of muscle fibers, there's a group of muscle fibers here, right? That's innervated by a single motor neuron. Here's a single motor neuron, is what we call a motor unit. Okay, the size of a motor unit can vary, and this will depend on the muscle. Some muscles, they contain motor units that innervate only a few muscle fibers, while others, they contain motor units that innervate many. When a motor neuron fires, all the muscle fibers that are innervated by that neuron contract together. So this means that the force that's generated by a muscle contraction depends on the number and size of the motor unit that are activated. So if a small number of motor units are activated, the resulting muscle contraction it'll be weak. Okay, it'll be relatively weak. While if we have a large number of motor units that are activated, the resulting muscle contraction will be stronger. So summation in physiology refers to the increase in force or tension that's produced by a muscle fiber or group of muscle fibers in response to repeated stimuli. It doesn't involve the adding together of individual twitch contractions to increase the intensity of the overall muscle contraction. Now, there's two types of summations that can occur in muscle fibers. Okay, there's temporal summation, or we also call it frequency summation. Okay, they're both the same thing. Temporal summation or frequency summation. This occurs when a muscle fiber is stimulated repeatedly before it had a chance to completely relax from the previous stimulus. So as a result, the force of contraction increases with each subsequent stimulus, and this leads to a sustained contraction that we call tetanus. Okay? Now, spatial summation, or it's also known as multiple fiber summation. This happens when multiple muscle fibers are stimulated simultaneously to produce a stronger contraction. And this is achieved by recruiting more motor units, which are groups of muscle fibers that are innervated by a single motor neuron. So wave summation is the addition of individual twitches in a muscle fiber, where the twitches are close together in time and the muscle fibers do not completely relax between them. Right? So this results in a greater overall force of the contraction compared to a single twitch. So again, remember, complete summation is also known as tetanus. Okay? And this is when the muscle fiber is stimulated at a high enough frequency that it doesn't have a chance to relax at all between the twitches. And this results in a sustained contraction. So going back to what we said, tetanization, this is a phenomenon in muscle physiology where a sustained muscle contraction is produced due to high frequency stimulation of muscle fibers. So when the frequency of stimulation reaches a certain threshold, the individual muscle twitches, they fuse together, and the muscle fiber is unable to relax completely between contractions. So as a result, the muscle contraction appears to be continuous, and this sustained contraction is known as tetanus or tetanic contraction. Now, tetanic contraction can be classified into two types: unfused tetanus and fused tetanus. Okay? Unfused and fused. In unfused tetanus, the muscle fiber is able to partially relax between contractions, resulting in a slightly wavering contraction. Now, in a fused tetanus, the muscle fiber is unable to relax at all, and this results in a sustained contraction. Now, the specific type of tetanus that occurs depends on the frequency and the duration of the stimulus. Okay? So both frequency and the duration of the stimulus is what will determine the specific type of tetanus that will occur. Now, even when muscles are at rest, they're not completely relaxed, but rather they exhibit a certain amount of tautness that we call muscle tone. So muscle tone is maintained by a low rate of nerve impulses that are constantly being sent from the spinal cord to the muscle fibers. These impulses are controlled by both signals that originate in the brain and by signals that originate in muscle spindles located within the muscles themselves. So, what are these muscle spindles? Muscle spindles, they're sensory receptors that are embedded within skeletal muscles, and they're responsible for detecting changes in muscle length and tension. When a muscle is stretched, the muscle spindle detects a change and sends a signal to the spinal cord, which in turn sends a signal to the muscle fiber, causing them to contract and resist the stretch. This reflexive contraction helps to maintain muscle tone. And muscle tone is important. Why is it important? Well, because it maintains posture as well as for stabilizing joints. And it's also necessary for proper muscle function during movement. Abnormalities in muscle tone can occur due to a variety of neurological or musculoskeletal disorders, and they can end up resulting in symptoms such as muscle weakness, spasticity, or even rigidity. Okay, so muscle weakness, spasticity, and rigidity are consequences of neurological or musculoskeletal disorders. All right, so muscle fatigue is a well-known state of tiredness and weakness that happens when a muscle is used strongly and for a long period of time. Muscle fatigue can also happen when the energy store has been depleted. Okay, so for example, depletion of glycogen. Okay, experiments have shown that the connection between nerves and muscles can weaken. Okay, they can get very weak after intense and prolonged muscle activity, making it harder for the nerves to communicate with the muscle. And this can further contribute to muscle fatigue. Additionally, muscle fatigue can also happen when the blood flow to a contracting muscle is interrupted. So this is because, as we know, blood carries nutrients. Blood also carries oxygen that muscle needs to function properly. So when blood flow is interrupted, the muscle loses its nutrient supply as well as its oxygen supply, okay, especially its oxygen, which then lead to muscle fatigue. All right, so the muscles in our body are constantly being remodeled or adapted to match the demands placed on them. This includes changes in muscle diameter, length, strength, as well as the vascular supply. Even the type of muscle fibers can be altered due to some extent to better suit the functions they're required to perform. Now, these adaptations occur in response to change in our physical activity levels, such as increase or decrease exercise, as well as in changes in our overall health and, again, our aging process. Okay, so look at these. Muscle hypertrophy, okay? Muscle hypertrophy refers to the process of increasing the size or mass of muscle, typically through strength training exercises that involve progressive overload. Now, the increase in mass is due to an increase in the size of individual muscle fibers, as well as an increase in the number of contractile proteins within those fibers. Now, on the other hand, muscle atrophy refers to the process of decreasing the size or mass of muscle that can occur as a result of not using that muscle or aging or certain medical conditions. During muscle atrophy, there's a decrease in the size and number of muscle fibers, as well as a decrease in the amount of contractile proteins within those fibers. And this may lead to weakness and loss of function in the affected muscles. Right, so after an individual dies, about 1 to 2 hours after death, the muscles of the body, they go into a state of rigidity called rigor mortis. And this happens even though there are no more action potentials. Usually this takes place at about a temperature of around 30 degrees centigrade. Okay, this is when that happens. Now, this rigidity happens because the muscles, they lose all their ATP, which is required to relax and separate the cross bridge from the actin filaments. So the muscles, they remain in a state of rigidity until the protein deteriorate. Okay? And this happens about 15 to 25 hours after, okay? After death. And again, this happens because of the release of proteolytic enzymes. Now, the speed of these events increases at higher temperatures. All right, now we're going to be going over transmission of impulses from nerve endings to skeletal muscle fibers. Again, we're looking at the events that take place at a neuromuscular junction, NMJ. Again, if you don't recall this, be sure you review your slides from Physiology 1. This, more or less, is it's a review of what you've already learned in Physiology 1. So be sure you review this. This is very important information. Okay, the skeletal muscles in our body are connected to nerves that carry signals from the brain to the muscles. These signals are also known as nerve impulses, okay, and they cause the muscles to contract and move. Okay. The nerve fibers that connect to the skeletal muscle fibers are large and myelinated. This means that they are coated in a protective layer that helps conduct the nerve impulses quickly. Now, these nerve fibers originate from large motor neurons that are located in the anterior horns of the spinal cord. Once the nerve fibers enter the muscle belly, they branch out and connect with several skeletal muscle fibers. Now, at the midpoint of each muscle fiber, there's a junction called the neuromuscular junction. Okay? This is where the nerve endings interact with the muscle fiber. So when the nerve impulse reaches the neuromuscular junction, it triggers the release of a chemical called acetylcholine. Okay? Acetylcholine is a neurotransmitter. Acetylcholine will bind to receptors on the muscle fiber and cause it to depolarize. The depolarization then leads to the muscle to contract. Okay? So, the action potential, which is the electrical signal that initiates the muscle contraction, travels in both directions towards the muscle fiber ends. So, at the neuromuscular junction, okay, where a nerve fiber connects, I shouldn't say connect, but it's in close proximity where it interacts with the muscle fiber. Okay, we have a complex structure that is made up of branching nerve terminals and invaginated membranes of the muscle fiber. The space between the nerve terminal and the muscle fiber membrane is what we call this space over here. It's what we call the synaptic space, or another term that they use for it is the synaptic cleft. Okay? So again, the space between the nerve terminal and the muscle fiber membrane, here's the muscle fiber membrane over here. All this. Okay? Here's the neuron over here. The space, this gap that's between them, is what we call the synaptic space, or another term is the synaptic cleft. And again, there are also numerous small folds of the muscle, as you can see over here. And we call these the subneural clefts. So together, these structures, they allow for the efficient transmission of nerve impulses to the muscle fiber, which leads to muscle contraction. So, as I said, acetylcholine is a neurotransmitter. Okay, and it's a very important neurotransmitter that's, in this case that we're looking at right now, is responsible for muscle contraction. It's produced in the axon terminals and it's stored in small vesicles. Okay, we call these synaptic vesicles. So when a nerve impulse arrives at the axon terminal, the vesicles release acetylcholine into the synaptic space. Then they bind to receptors on the muscle fibrous membrane. So this binding triggers a series of events that leads to muscle contraction. Now with that said, acetylcholine only stays in the synaptic space for a very short time because we have another enzyme there, okay, that recycles it. So that enzyme is called acetylcholine esterase. Acetylcholine esterase, again, it's an enzyme that breaks down acetylcholine and sends it back to the axon. of the neuron, and then it will get reused. So, when a nerve impulse reaches the neuromuscular junction, it triggers the release of synaptic vesicles that contain acetylcholine from the nerve terminal into the synaptic space. This is what you see over here. Okay? So these vesicles merge with the nerve membrane and release that acetylcholine into this synaptic space. And now the acetylcholine will come and bind. Okay? What does it bind to? It binds to receptors that are located near the mouth of these subneural clefts on this motor end plate, or we call it the muscle membrane. So these receptors here are called acetylcholine gated ion channels. Okay, acetylcholine gated ion channels. Why? Because these are proteins that are ion channels. But again, these are a specific type. First of all, these are referred to generally as ligand-gated proteins. And these ligand-gated protein gets named according to the ligand that they interact with. The ligand in this case would be the neurotransmitter, and the neurotransmitter is acetylcholine. Therefore, it is acetylcholine-gated ion channels. So binding of acetylcholines opens up these channels, and this is going to allow for charged ions like sodium to come rushing inside this muscle fiber, which will then initiate an action potential, okay, in this muscle fiber. Okay, Myasthenia gravis is a rare condition that affects about 1 in 20,000 people. It causes muscle paralysis due to the inability of the neuromuscular junction to transmit enough signals from the nerve fiber to the muscle fibers. Researchers have discovered that most patients with myasthenia gravis, they have antibodies in their blood that attack the acetylcholine receptors, which are essential for muscle contraction. So what does this suggest? This is saying that myasthenia gravis is an autoimmune disease, okay? Meaning that the immune system attacks the body and ends up destroying the cells and tissues. So in this case, the immune system mistakenly targets the acetylcholine receptors at the neuromuscular junctions, leading to muscle weakness as well as paralysis. All right, so the disease usually affects women in the 30s, and it's characterized by symptoms such as fatigue, drooping eyelids, okay? We call that ptosis, and also double vision, diplopia. Symptoms, they tend to improve with rest, in addition to using anticholinesterase drugs. All right, moving on to smooth muscle. Right, so smooth muscle is indeed composed of smaller fibers than skeletal muscles. And we have two main types of smooth muscles. There's multi-unit smooth muscles and unitary smooth muscle, or single unit smooth muscle. That's another name for it. Unitary or single unit smooth muscle, both the same thing. Okay. So now let's talk about multi-unit first. So multi-unit smooth muscle consists of individual fibers that are innervated by nerves and contract independently of each other. Okay. Now this type of smooth muscle is found in structures such as the iris of the eyes, the erector pili muscles of our hair follicles that we find within the skin, the walls of larger blood vessels, and also even around the pupils. Okay. Now, unitary smooth muscles, on the other hand, they're made up of many cells that are electrically coupled to one another and contract as a single unit. Now, this type of smooth muscle is found in the walls of most hollow organs, like your stomach, your intestines, and in women, the uterus. Now, multi unit smooth muscles are made up of discrete, separate smooth muscle fibers. Now, these fibers, they operate independently of one another, and they're often innervated by a single nerve ending. Multi-unit smooth muscles, they're found in organs where fine control is needed, such as the iris and the ciliary muscles of the eye, or even in the airways and blood vessels where the diameters need to be regulated carefully. Okay, unitary smooth muscles are also known as synctial smooth muscles or visceral smooth muscles. This is a type of smooth muscle that forms a mass of hundreds to thousands of smooth muscle fibers that contract together as a single unit. Now, in this type of muscle, in smooth muscle, you will not find troponin. The troponin complex ends up being replaced by calmodulin. Calmodulin is the protein that's replacing the troponin complex. Now, where do we find unitary smooth muscle? We find it in the walls of most of the viscera of our body, including our GI tract, our gastrointestinal tract. We find them in bile ducts, ureters. In women, they're found in the uterus. And also we find them in many blood vessels. Unitary smooth muscle is characterized by the presence of gap junctions, which allows the muscle fibers to communicate and contract in a coordinated manner. Now, keep in mind, gap junctions are also found in cardiac muscle as well. Okay? So we don't find gap junctions in skeletal muscles. So, unitary smooth muscles, they also exhibit spontaneous contractions that are known as slow waves or pacemaker potentials, which can be modified by neural as well as hormonal inputs. Okay? So, again, one of the reasons this is able to take place is because of the presence of the gap junctions. Unitary smooth muscles will contract slowly and rhythmically, and they're able to maintain a sustained contraction for a prolonged period of time. And this makes them well suited for the many roles that they have in many visceral organs. In smooth muscles, actin filaments are attached to dense bodies that are dispersed throughout the cell, and myosin filaments are scattered among the actin filaments. The dense bodies serve as attachment points for the actin filaments, which are not arranged in a striated manner that we see in skeletal muscle. On top of that, smooth muscles lack the troponin complex. Right? Remember what I said. We don't have troponin complex. Instead, there's calmodulin. Okay? Calmodulin. So, calmodulin is what's taking over the role of troponin, which we find in cardiac and skeletal muscle. Okay, so again, instead of troponin in smooth muscle cells will have calmodulin. Calmodulin is a protein that binds to calcium, which then activates the myosin light chain kinase and triggers the smooth muscle contraction. Smooth muscle contraction is activated by an increase in the intracellular calcium ions. And most of the calcium ions that cause contraction enter the muscle cells from the extracellular fluid. Now, ATP is also required for smooth muscle contraction, but the energy requirement of ATP is lower than what we find in skeletal muscles And this is because of the slower cycling of cross bridges in smooth muscles. So this slower cycling also contributes to the prolonged duration of smooth muscle contractions compared to the skeletal muscle contractions. And that concludes this week's lecture. Thank you again, and we'll see you guys next class.